Hi everybody, and welcome to our module on polymerase chain reaction. Polymerase chain reaction, or PCR, is a laboratory technique whereby DNA molecules in a sample are amplified or copied to make multiple copies of DNA. And this has a number of uses. For example, you can make more DNA from a very small amount in a sample. In addition, you can use PCR to determine if DNA is present by seeing if it amplifies via this technique. And then finally, you can also determine the amount of DNA by seeing how quickly it amplifies. If you start with lots of DNA, it will amplify rapidly. If you start with very little DNA, it will amplify slowly. And here's a picture at the bottom of the screen just to keep in your mind of what PCR does. It takes a double-stranded DNA molecule and converts that into multiple copies of that double-stranded DNA molecule. The way PCR works is that you put a sample containing the DNA of interest into a tube with a number of other ingredients, which will then be heated and cooled over and over in cycles. The other ingredients you need in the tube include DNA polymerase, which is the enzyme that will copy DNA. And then, as you may know, if you've watched the videos on DNA replication, DNA polymerase requires a primer. In other words, it requires a short stretch of nucleic acids in order to begin making more DNA. So you have to put a primer in with the sample. That primer is a single-stranded DNA segment, and it's complementary to the DNA that you want to study. So for example, if you wanted to amplify DNA that was complementary to this short nucleic acid sequence I've shown on the screen here, this is the primer that you would use. And then finally, of course, you need to add nucleotides, which will be used by DNA polymerase to make more DNA. The way that PCR works is you put all those ingredients into a tube and then you heat and cool the tube. This is called a cycle. And after each cycle, the amount of DNA in the tube will double. So the first step in a PCR cycle is to heat the sample. This denatures double-stranded DNA into single strands. You then cool the sample and the primer anneals or binds to the complementary DNA if it is present. Then you warm the sample and DNA polymerase will elongate from the primer and make new DNA. And you repeat this process in cycles, and each cycle generates more DNA. So here's a picture from the web just to make this very clear. On the left side of the picture, we have our initial ingredients. The green structure is the original DNA to be replicated. The red structure is the primer, and the blue is the nucleotides. We then heat the sample and the DNA denatures, as we see right here. We then cool it slightly so that the primer anneals. We then warm it so that DNA polymerase begins elongating the strands. And you can see here that now we're going to get 2 DNA strands where we began with only 1 DNA strand. We then repeat that process in another cycle and we go to 2 to 4, and then we can repeat it again. We'll go from 4 to 8. And you can see that how you very rapidly get lots of DNA even from a tiny amount in an initial sample. Now, it's easy to imagine from that last picture how a small amount of DNA can be amplified by PCR, but another use of PCR is to quantify the amount of DNA in a sample. This is done by a variant of PCR called real-time or quantitative PCR. In this case, PCR is done in the presence of a fluorescent dye, and the amount of dye that's taken up is proportional to the amount of DNA in the sample. So if there's more DNA, there is more fluorescence. So the way this is done is the amount of fluorescence is detected as PCR is ongoing. If there's a rapid increase in fluorescence, this means the initial sample contained more DNA. If there's a lesser rise in fluorescence over time, it means the initial DNA sample had a relatively small amount of DNA. PCR has lots of uses in research, but I've listed on the slide some of the clinical uses of PCR. And the thing you want to remember about these clinical uses for step 1 of your boards is PCR is used in situations where you have very small amounts of DNA, usually that is not part of a chromosome. So this is usually used to seek out and identify small amounts of DNA that might be circulating in tissues or plasma. So one use along those lines is to diagnose herpes simplex virus encephalitis. Herpes simplex is a DNA virus, and in patients who have encephalitis from this virus, there will be small amounts of herpes simplex DNA in the cerebrospinal fluid, and you can use PCR to amplify those small amounts of DNA and make the diagnosis. PCR can also be used to measure an HIV viral load. Now, this may seem confusing to you because you may know that HIV is an RNA virus. However, to use PCR for viral load measurement, the enzyme reverse transcriptase is used to make a copy of the virus's RNA. This is called cDNA. And then you can use PCR to amplify the cDNA, and the amount of cDNA produced over time indicates the viral load, as we just discussed for quantitative PCR. And this is one of the standard tools for monitoring viral load in HIV patients. And that concludes our module on PCR.